All right, welcome. Today, we're putting on our detective hats and diving into a case that you are absolutely going to see in your practice and on your exams. We're talking about microcytic anemia, and we're going to crack this case wide open. So, let's set the scene. Your patient comes in, they're tired, they're dizzy, you know the drill. You do the right thing, you order some initial blood work, and the first report hits your desk. This is where the real investigation starts. And right there on the report is our first major clue. The red blood cells are small. So, what now? What's your next move? That's the exact question we're going to answer right now. Okay, first things first. Before we can solve the puzzle, we have to understand what the pieces mean. What are we actually talking about when we say microcytic anemia? Let's lay down the fundamentals. So, anemia is kind of a broad term, right? At its heart, it just means your blood isn't carrying enough oxygen. Maybe you don't have enough red blood cells, or maybe the ones you have just aren't up to the job. Now, the microcytic part, that's where we get specific. It all comes down to one key number on that lab report, the MCV, or mean corpuscular volume. It's just the average size of your red blood cells. And when that number drops below 80 femtoliters, boom, that's our official diagnosis. The cells are microcytic. They're tiny. And this table here really helps put things in perspective. See, we classify anemias by cell size. You've got your normal size cells, or normocytic. Think of something like acute blood loss. Then you have your big cells, macrocytic, like you'd see in a B12 deficiency. But our investigation today, we're focused squarely on that top row, the small microcytic cells. So the big question is, why are the cells small? It's not random. A small red blood cell is a huge clue that points to a specific type of problem, a problem on the assembly line. It means something's going wrong during production. And what are we trying to produce? Hemoglobin. That's the protein that makes red blood cells, well, red. More importantly, it carries oxygen. Think of it like the stuffing inside a pillow. If you don't have enough stuffing, you can't make a full-size pillow. Same idea here. Not enough hemoglobin, you get a smaller microcytic cell. Simple as that. Now, this production problem really boils down to two main things. Either you've got a problem making the heme part, that's the bit that contains iron, or you've got a problem making the globin part, which are the protein chains that hold the heme. Every single type of microcytic anemia we're going to talk about fits into one of these two buckets. Okay, so we know the how. Now let's meet the who. When you see microcytic anemia, there are four main suspects you need to round up for questioning. Let's get our lineup ready. Exactly as we laid out. Under the heme problem category, we've got three culprits. Iron deficiency anemia, that's the big one you'll see most often. Anemia of chronic disease, and the more unusual, sideroblastic anemia. And over in the globin problem corner, we have one major player, thalassemia. Now, before we even think about ordering more advanced lab tests, the patient themselves is a treasure trove of clues. Your history and physical exam are your first and best tools for gathering evidence and starting to figure out which suspect is the most likely. First off, you're going to see the general, run-of-the-mill symptoms of any anemia. It makes sense, right? If you're not delivering oxygen well, you'll get tired, feel short of breath, look pale, your heart might beat faster to try to compensate, and you can feel dizzy. These are the classics. But those are too general. We need to be better detectives. We're looking for the really specific clues, the high-yield signs that let you start pointing fingers at one diagnosis over another right there in the exam room. And for iron deficiency anemia, you've got some absolute textbook signs. If you see these, your ears should perk up immediately. Koilonychia, which are those weird spoon-shaped nails. Pica, that strange craving to eat non-food stuff like ice chips or even dirt. And things like cracks at the corners of the mouth called angular cheilitis. These are dead giveaways. And here's another huge one, but this time it's a clue you find under the microscope. If the lab report mentions basophilic stippling, these tiny blue dots scattered inside the red blood cells, a giant alarm should go off in your head for sideroblastic anemia. It's a classic buzzword, especially for lead poisoning. All right, we've gathered our physical clues, we've got our suspicions. It's time for the final act, the lab test showdown. This is where we use cold, hard data to separate our suspects and definitively crack the case. A really great first sorting tool is the RDW, the red cell distribution width. All it tells you is how much variety there is in the size of the red blood cells. So if you have a high RDW, it means you have a real mix of sizes, big ones, small ones, all over the place. That points you toward iron deficiency or sideroblastic.
If the RDW is normal, it means all the cells are pretty much the same size. They're just all uniformly small. That suggests thalassemia or anemia of chronic disease.
But this is the main event, the iron panel. This is how you nail the diagnosis. Each of our suspects leaves a unique fingerprint on these tests. In iron deficiency, almost everything's low, except for the TIBC, the body's capacity to bind iron, which is sky high because it's desperate for more. In anemia of chronic disease, the body is actually hiding iron away, so serum iron is low, but ferritin, the storage iron, is high. And in thalassemia and sideroblastic anemia, you're often dealing with iron overload, so everything looks high. See? 4 different patterns.
And what if you really suspect sideroblastic anemia? There's one final definitive test. You get a bone marrow biopsy and you use a special stain called Prussian blue. If you see what's called ringed sideroblasts, that's the smoking gun. It's literally a picture of iron getting trapped in the mitochondria around the nucleus, unable to be made into heme. Case closed.
Okay, pop quiz. Let's put this all together. Your patient has a low MCV. You get their iron studies back. Serum iron is low, but their ferritin is high. Based on what we just saw, which diagnosis shoots to the top of your list?
The answer? It's got to be anemia of chronic disease. And that high ferritin is the absolute key. If it were iron deficiency, the storage form of iron, ferritin, would be in the basement. But in ACD, the body has plenty of iron. It's just locked it away in storage because of inflammation. That's why the ferritin is high. Nailing that one difference is often what it takes to get to the right answer. And just like that, you've gone from a single clue to cracking the entire case.